Professor Dan Roden, I should introduce as Professor of Medicine, Pharmacology and Biomedical Informatics and Senior Vice President for Personalised Medicine at Vanderbilt University Medical Centre. He's a very active and internationally renowned researcher in clinical pharmacology and cardiology, and he's led world-leading programs in pharmacogenomics, including Emerge and Predict, which uses electronic health records for preemptive pharmacogenomics and for delivering high-quality health services. A very warm welcome to you, and can I invite you to give us this year's forum lecture. Thank you very much.
So I will, thank you so much for the invitation. Thank you to you and to Munir Pir Mohammed, my good friend for a long time, and one of the engineers of my being here. So this is the topic that I was actually assigned, but if I had not been assigned it, I probably would have chosen it anyway. I do love this graphic because it has people in it, in their DNA, and it has outliers. And we're going to talk about population pharmacogenomics, and we're going to talk about outlier pharmacogenomics. Colin Dollery, I met Colin Dollery on multiple occasions. I was telling Munir this story last night, and if Lady Diana is online, she may remember also that she and Sir Colin and I were in a meeting in Kuala Lumpur, and we flew from Kuala Lumpur to Hong Kong in a typhoon. A memorable trip because we bounced around a lot. He really was one of the 2 or 3 founders of the discipline of clinical pharmacology. I had the pleasure of working with his counterpart in the United States, John Oates, who was a contemporary, and between the two of them, you couldn't do better. We're here. I like this mission statement, vision statement, making medical science work for everyone. I've reflected often in my advanced years on a career that has done a lot in discovery, and I'll show you some of that. But at this point, what I wish is that we would take some of that discovery and actually use it to make healthcare better for people. So that this really resonates with me. And then the personalized prescribing report that Munir and others generated is a blueprint for that for pharmacogenomics, and I'll come back to that toward the end of the talk. So this is my view of the spectrum of pharmacogenetic discovery and implementation, all the way from basic biology and basic biologic discovery all the way through implementation. And I'll talk mostly about the right-hand side of this slide during the talk, but let me start with a little bit of biology. These are old data, old data, and you can display this kind of data for virtually any drug. And the idea is that if you give a dose of a drug to a large population of patients, you will have a normally distributed set of responses. Most people are at or near average. Some people are away from the average. That's what a normal distribution is. And people don't realize that, but there's variability in response to virtually any drug that we ever choose to use. And that's part of the problem of pharmacogenomics. This is from the Pharmacogenomics Research Network, a network that the NIH in our country sponsored for about 20 years and now exists as a freestanding entity. At some point, I should give a long discussion about the difference between pharmacogenetics and pharmacogenomics, but I don't have time for that. I'll just say that genetics has this implication of 1 or 2 variants, and genomics is the whole genome. We'll get there. So this is the other part of variable drug responses, and that is the idea that there are rare, very serious adverse drug reactions. Some of them are shown here. I'm not going to talk about any of them in detail, but just to show that these things exist, and they are huge problems in clinical medicine, often viewed as idiosyncratic because we don't have a good explanation for them. But at the end of the day, each one of them does have an explanation. If we understand the underlying biology, perhaps we'll be able to understand how to avoid them. I will talk a little bit more about the Stevens-Johnson syndrome later, but just to say that all you have to do is see one patient with Stevens-Johnson syndrome to convince yourself that it would be a great thing not to ever see a second patient with Stevens-Johnson syndrome ever again, because this is a potentially fatal or often fatal skin and other organ disease. So in the United States, the National Academy of Medicine, at the time was the Institute of Medicine, issued a report in the 1990s called To Err is Human. And their estimate was that there are over 2 million adverse drug effects in hospitalized patients in the United States, that it was the 4th to 6th leading cause of death among hospitalized patients in the United States. 10 years later, there was a review in the New England Journal of Medicine, and there was zero evidence that anything had changed. So 100,000 people a year is the equivalent of 2 jumbo jets running into each other every day. Now, if that happens once in a decade, the regulatory authorities get very, very excited about that. Yet we have this gigantic problem. We think some of it is avoidable, and we're not doing enough about it. So why is it that there's variability in drug response? A clinical pharmacologist has many versions of this slide, but I chose to use the one from the personalized prescribing report because I think it makes things simple. So there are many, many reasons why we have variable drug responses. Underlying diseases, drug-drug interactions, drug-food interactions, non-adherence, patients' inability to understand which medicines to take when. We often blame the patient because they are not compliant. But compliance has this term that sort of somehow we are given an order and they have to follow it, whereas adherence has this sense that we're part of a care team, and often people don't take their medicines because they just don't understand how to take them or when to take them. And then, of course, there's the problem of genomic variation. So that's one of many reasons why people will have variability in response to drugs. So let me talk a little bit about the history of pharmacogenomics. Probably starts with Garrod, who is famous for describing the term inborn errors of metabolism, the idea that we all have metabolites that are in our body and we biotransform them into other metabolites all the time, endogenous metabolites. But he also speculated in chapter 1 of this book that there would be also odd variable drug responses because of variable responses to drugs because of a similar kind of chemical basis, he called it. Of course, he didn't ascribe it to genes, but he had this sense that there were chemical biotransformations occurring in the human body. So in the 1940s and 1950s, there were a bunch of pretty spectacular examples of abnormal drug responses that were generated. They're listed here. I'm not going to read them for you. And then in the 1950s, Johns Hopkins was a center for study for variable drug responses. One of the centers in the world, in the country, in the country and in the world. And the chairman of medicine at the time, Victor McCusick, or he became chairman of medicine, is one of the drivers of the discipline of clinical genetics and pharmacogenetics. And one of the very first pharmacogenetic phenomena that were well described was McCusick working with a fellow, and that fellow was David Price Evans, who was chairman of medicine here for many years, and is, at least in my world, famous for describing variable acetylation as a contributor to variable responses to anti-tuberculous drugs and many other drugs now. So Munir, earlier in his career, looked at adverse drug reactions as a cause of admission, and looked across over 18,000 patients, and 6% of all admissions were associated with an adverse drug reaction. So it gives you a sense of the scope of this problem. The slide just lists the drugs. I'm obviously not going to go through them, but somewhere around a quarter have a prominent genetic etiology. So there is an opportunity to intervene if we understand the genetics and understand which patients are at risk because of their genetics. My introduction, this is really ancient history, my introduction to pharmacogenetics occurred when I was a fellow, when I think Munir was in high school or something, or probably in grade school. So I came to Vanderbilt, and I was interested in cardiology, but I came as a clinical pharmacology fellow, and I studied a drug called Enconide, an antiarrhythmic, and I hope none of you have ever used it. It has prominent pro-arrhythmic effects in some patients. But this is early in the clinical pharmacology, and one of the things we did was we gave everybody, we had 11 patients in our trial, we gave everybody a single 25 milligram dose and followed plasma concentrations for a day and then dosed patients up chronically. drug was given 3 times a day. So we had 11 patients. 10 of them had this amazingly gratifying response. I won't go into the details, but they really had a great pharmacologic effect, we thought. They're characterized in blue by a very rapid drug elimination and tremendously variable concentrations. You can see the concentrations vary over 2 orders of magnitude. And then we had one outlier, and the outlier was, we expected the outlier to have low plasma concentrations. She in fact had very, very high concentrations and very, very slow drug elimination. So I worked with a bunch of very, very smart pharmacokineticists at the time, and we spent months trying to sort out why this happened. When I present it this way to people now, everybody looks at it and says, oh, I understand that. And I'll come back to the explanation in a second. But it took us a long time. And then we had a visitor from the UK named Bob Smith, who was from St. Mary's. And he was studying a drug called debrazequin. Debrazequin is an antihypertensive. And he made the mistake, we think it's a mistake, perhaps not, he was investigating the clinical pharmacology, so he had all the fellows in his lab take a dose, and he took a dose. And he was profoundly hypotensive on a single dose that didn't affect other people. So the mythology, of course, is that had it been a fellow, he would have ignored it, but because it was him, he spent a lot of time investigating the mechanism underlying this. So it turns out debrazequin is an antihypertensive and is biotransformed to a 4-hydroxy inactive metabolite. And he was lacking in the ability to biotransform, so he had very high plasma concentrations of the parent drug, very big pharmacologic effect. So it turns out enconide is a prodrug. Enconide is not the antiarrhythmic. It is biotransformed to its major metabolite to generate its effects, and that explains why the poor metabolizer has no pharmacologic effect. The rapid metabolizers do. And it turns out that debrazequin and enconide use the same pathway. And the enzyme that does that is an enzyme called CYP2D6, and it accounts for the biotransformation of about 25% of the drugs that we use chronically. And I remember he gave this seminar at Vanderbilt, and we walked out of the room, and my mentor and I looked at each other and said, well, we have got to get some debrazequin and test it on our enconide patients. And it turned out that's how we made this discovery. So if you look at CYP2D6 biotransformation activity across a population, it falls, this is a Swedish population, but it falls in several hundred subjects. It falls into two normal distributions with separation between the two. So the way this axis works is that if you're on the right, you have little activity of the enzyme, and if you're on the left, you have lots of activity of the enzyme. And so you can see that there's an anti-mode indicated by the heavy arrow. And you can see that there's variability in response even among the extensive metabolizers. And the way this works, we understand now genetically, is that poor metabolizers are heterozygotes or compound heterozygotes for loss of function alleles. And then the blue group has 2 wild-type alleles, or perhaps only 1. It's hard to tell the distribution between those two, so they tend to overlap. And then as time passes, individuals who have multiple copies of the gene that are functional, so they make lots and lots and lots of enzyme, are also identified. And they're at the left end of the distribution, as you can see. So there are many loss of functional alleles, and some of them exist in some populations, but not in others. And we'll come back to that theme, because that's one of the themes in pharmacogenomics and in genomics in general these days. So Mike Steen, a very talented clinical pharmacologist, and I proposed this idea of high-risk pharmacokinetics. And there are 2 scenarios that we had proposed. One was the enconide scenario. You give a prodrug, the patient is unable to metabolize the prodrug, and so they get no pharmacologic effect. And enconide is an example. I'm going to talk a lot about clopidogrel, the antiplatelet drug that's widely used in patients with coronary stents, and then tamoxifen and codeine are other examples, which I'm not going to talk about today. The other scenario is the debrazequin scenario. The parent drug is active and has only one pathway for biotransformation. So many drugs have 6 pathways for biotransformation. And if you're lacking in activity for one of them, it doesn't really matter. But debrazequin only has 1. And so if the pathway is absent because of genetic factors or because of co-administration of an inhibiting drug, you can then get exaggerated effects. So debrazequin, warfarin, irinotecan, azathioprine fall into this category. And I'll talk a little bit about azathioprine. I'll give you one little example of azathioprine later. The other way that the second scenario can happen is if the drug is excreted by the kidneys. There are many drugs that are excreted unchanged, and of course, if you develop renal failure, you will, you'll get that effect. And I'm actually not going to talk about sotalol later. And then that implies that all adverse drug reactions are based on pharmacogenetics, how you metabolize. And that's a dramatic oversimplification. There are many, many adverse drug reactions that are unrelated to the plasma concentrations, but related, in fact, to other factors that are genetic, largely, or some of them are genetic. Stevens-Johnson syndrome is an example of that. So this is much of the biology or the basic science of clinical pharmacology or pharmacogenetics as I want to talk about today, because I want to talk about the implementation piece. I came to Vanderbilt because I wanted to study clinical pharmacology, and I got involved in the idea of pharmacogenetics, and Vanderbilt was a major leading center for that kind of science when I came, and to this day still is. The other thing, I take care of patients, and so when I came, we, like everyone else, had a paper medical record. This is a picture of our medical record room, but we converted in the 1990s to an electronic medical record system. Our leadership made an institutional commitment to the discipline of biomedical informatics, and now there are over 150 faculty in the Department of Biomedical Informatics at Vanderbilt. It's one of the largest departments in the world in that space. So between the idea of focusing on variable drug responses and the idea of this clinical bioinformatics effort, we were in a great position to create resources for personalized medicine. So we made a strategic plan, like other people do, and personalized medicine was one of the hallmarks of that strategic plan. Notice I spell it with a Z here, or a Z, as opposed to an S, but I hope you'll forgive me. So if you do a PubMed search for Vanderbilt and personalized or precision medicine, this is the number of references that you get. I just updated this a couple of days ago, and I'm very proud of the fact that the very first one is this paper that describes the development of our biobank. Now, I can talk about our biobank for a couple of hours if you wish. We've named it BioVU because the VU stands for Vanderbilt University. But it's become important as a tool for developing precision medicine in many, many spaces. So that's why I feel an urge to say it. So this is a quotation from Francis Collins, who was at the time the newly appointed director of the National Institutes of Health in Washington. And he was interviewed by the New England Journal of Medicine. And he was asked about pharmacogenomics. And this is all he said about it. That's all you need to say. So Francis was a practicing genetic clinician, as well as being a brilliant scientist and administrator. And he made the observation that the limiting factor is that you write a prescription, and then you get a genetic test, and then you wait around for a while. A week is what he said, but maybe it's a couple of hours, and maybe it's a couple of weeks. And then you can tell the patient whether you got the dose right or whether you got the drug right. And that just seems sort of excessively cumbersome, and that's one of the reasons that people haven't implemented pharmacogenetics. And then, of course, he said it's, once everybody's genome is sequenced, it's going to be easy and it'll improve outcomes and the world will be a better place. And I'm fond of saying when I show this slide that I agree with everything except that word, because it's anything but simple as we've learned. So making the case for preemptive pharmacogenetic testing involved a large study in BioView, following patients who had been at Vanderbilt for a long, long time. So we have 3.5 million electronic medical records, but some of those patients touch the system, and some of those patients come back year after year after year after year. So we looked at the medical home group, a subset of the medical home group, in fact, and asked the question, how many people get a drug that has a well-recognized pharmacogenetic story around it? And the answer was that 65% received at least one medicine within the previous 5 years. And Jonathan Schildkraut, now a senior member of our biostatistics faculty, made the estimate that had we been able to mitigate those adverse reactions due to those drugs, we would have mitigated probably 400 out of 50,000 people. So it's a number. It's not a huge number, but it's still an important number when you think about this rare adverse drug reaction problem. So we generated a program that we call PREDICT. That's what PREDICT stands for. And the idea is really encapsulated by the data I've just shown you, by Francis Collins' quote and by this cartoon in the New Yorker. This appeared on the occasion of the unveiling of the first draft of the human genome at the end of Bill Clinton's presidency, when Francis Collins and Craig Venter were in the East Room of the White House. And I'm fond of saying when I show this cartoon that she's handing the pharmacist her sequence on a piece of paper. So we're pretty sure that that's not the way it's going to work. She's not handing it to a physician. She's handing it to a pharmacist. So this is a team sport, very much a team sport. And she wants him to do something good with it, and he has no idea what to do with it. So there's an educational imperative that we recognize and we're meeting, but we still have a long ways to go. So what we did in PREDICT at the beginning was we spent a huge amount of time reviewing the literature. And then we would look at what the professional society said and what the FDA labels for various drugs would say. And then we would actually replicate known pharmacogenetic findings in BioView, and we did that. And then once we had a drug that we wanted to implement, we would take it to the Pharmacy and Therapeutics Committee. They would look at the evidence. They would always say yes. I shouldn't say it that way, but they did. And then we would implement using our biomedical informatics engines, including automated decision support, which I'll show you an example of in a second. So at the beginning, we took a program to an internal review process, and we had a very elaborate program. And the internal reviewers, who were people in general internal medicine and general pediatrics who didn't know pharmacogenetics but were sort of fans of this idea, said, this is very complicated. Why don't you just implement CYP2C19 and clopidogrel only in homozygotes, so a very small population, as I'll show you in a second. And we thought that was boneheaded, but we did it that way, and it was a good idea, just to sort of figure out how to so start slow and get it to work. But at the end of the day, after about a year, we had 5 drug gene pairs going. So time has passed, and we don't do the internal evidence review anymore. We rely on the Clinical Pharmacogenetics Implementation Consortium, a group of people with whom I've been involved, and many people across the United States have been involved, whose mission is to review evidence and say, if you had genetic information, how would you act on it? And the CPIC spends a lot of time not telling people whether they should get genetic testing, but just saying, this is what you should do with the data if you have the data. It's a little bit circular, but that's the way they look at it. So I have to tell you why we implemented CYP2C19 at the beginning. This is one of my mentors, Grant Wilkinson. Grant was from Leeds or Manchester?
Near Chesterfield.
Where?
Belpa.
I don't think that's true. Yeah. Well, Grant was from many places. Sometimes when I talk about Grant, I actually show his CV. And his CV lists his name, his date of birth, and his London club, yeah, the Athenaeum. Anyway, that's a slide I took out. So in the early 1980s, he was working on a drug called mefenitoin, a drug no one in this room has ever heard of, except perhaps in this context. And he described a polymorphism in the metabolism of mefenitoin. So he identified a group of poor metabolizers in whom you could not find the major metabolite in the urine, and a group of normal metabolizers. It's about 2 or 3% of the population he studied were poor metabolizers. So I gave him a very hard time. I said, Grant, why are you studying a dead drug? Because no one's interested and no one could care less. Well, it turns out that, fast forward, we're designing PREDICT, and the FDA adds pharmacogenetic, a black box pharmacogenetic label to clopidogrel, Plavix. And the black box says, look at the bottom bullet point, alternative treatment or treatment strategies in patients identified as CYP2C19 poor metabolizers. It didn't say you have to do it. It just said, if you happen to know, then this is the way to do it. So of course, clopidogrel is like Enconide. If you give it to patients who are poor metabolizers, they will not have an antiplatelet effect, and they're high at a risk for in-stent thrombosis. So it's actually an example of a major adverse drug effect being failure of the drug to do what it's supposed to do. And I think that's actually the commonest adverse drug effect there is. So, and of course, S-mefenitoin hydroxylase is CYP2C19. So Grant was right. Basic science can inform the way we take care of patients in ways that we often don't understand. So this is the way the PREDICT data are now displayed in the electronic medical record. I think it's fair to say that this is me. It doesn't say that anywhere, but this is mine. And it's pretty uninteresting, actually. But the point is that if I were to be prescribed Plavix and I'm a poor metabolizer, this pop-up appears automatically and says, are you sure you want to do this? Here's why you shouldn't do it. It's in red. And so that's the way the system works. We looked at the first 15,000 or so patients who had been genotyped, and only 2.7% were homozygotes for the loss of function alleles. The alleles that we survey are those 5 alleles that are listed at the bottom. Star 1 is normal, so these are all variants that are loss of function. And then 20% are heterozygotes. So they're at modest risk for stent failure. And then the others have no variant. So you look at these data in your cardiologist and you say, well, 2.7%, and there's such variability in response anyway. Why should I genotype all my patients? And that's the resistance that we get from the clinical practicing cardiologists. So when we looked at our first 10,000 PREDICT patients across all 5 drug gene pairs, what we found was that almost everybody has a variant that you'd like to know about. You don't know what gene the variant's in, and you don't know that they're going to get the drug, but everybody has a variant. They're that common. And then the other thing that we and many others have noticed is that if you look in large data sets, there's a data set called NOMAD that is searchable online, you can find that there are literally hundreds of variants in CYP2C19 that are missense, so they change the amino acid, and we have no idea how many of those are actually loss of function. They're rare. But you know that some people who are failures of clopidogrel response are failures because they have these rare variants that we don't assay because we don't know their function. So I'll come back to that as a problem or an opportunity. The other thing about CYP2C19 that is actually pretty interesting is that, so star 1 is the normal, and you look across the board, and most people are normal except for Pacific Islanders, who are mostly abnormal. Star 2 is the one that everybody pays attention to, and it's sort of somewhere between 12 and 61% of people carry star 2, very prominent in Pacific Islanders. And then star 3 is 9% in East Asians, 15% in Pacific Islanders, and absent everywhere else. So is that important? Well, the state of Hawaii sued Bristol Myers Squibb and Sanofi, the makers of Plavix, because they said the label didn't recognize that it wouldn't work in their population. Pacific Islanders and East Asians are highly represented in Hawaii, and they won. They won $1 billion in a lawsuit. Now, whether that stands or not, and whether that's the way to implement pharmacogenetics, I'll leave to I'm not sure we want the lawyers to be the ones telling us how to do this. Certainly in the United States, this is one way forward in terms of implementing pharmacogenetics. We've looked at a little bit of outcome data. So one question was, when clinicians get the data, do they act on it? Well, part of it is, do they get the data the day after, or do they get the data 3 weeks after? So that's a story. But so this is how many people are still on clopidogrel at 3 months. So if you're a normal metabolizer, almost 100% still are, as they should be. If you're a poor metabolizer, only 40%. So that's good. I mean, it should be 0, but at least it's not 100%. And what's interesting is that the ones who have one loss of function allele are in the middle. So clinicians are smart enough to know that it's the poor metabolizers that you really have to pay attention to, and the others, not so much. There are many reasons why somebody wouldn't switch. The other medicines are more expensive, they have bleeding complications, that kind of thing. So we couldn't expect 100% switch over, but those are the data. And the other factor that predicts switching is this, which sort of, it's not really an embarrassment, but there it is. These are, so the estimated proportion of patients who are on alternate antiplatelet therapy at 12 months, in patients who are normal, those on the left, who don't have variant alleles, it's about 10 to 20%. And in patients who have abnormal alleles, it's more than that or on alternate therapies. And each dot, or each pair of dots, represents one cardiologist. So some cardiologists pay lots of attention, and some cardiologists pay almost no attention. They all pay a little bit of attention. So inter-prescriber variability, something we probably need to do something about. So let me talk a little bit about Stevens-Johnson syndrome just for a second, because there's a story here as well. 15 years ago, Munir, you can tell me when this happened, a group in East Asia identified HLA-B star 1502 as a susceptibility allele for Stevens-Johnson syndrome. Very powerful susceptibility allele, as one of our speakers will tell us later. So in Hong Kong, where that is the prevalent allele, they did an experiment where they said everybody who goes on carbamazepine, Tegretol, should get genotype beforehand, and if they have the variant allele, they should get something else. So that seems like a reasonable idea, and it seems like it should work. So new prescriptions for Tegretol fell from 16% to 2%. So doctors said, well, I'm just not going to do this. It's just too much of a pain in the rear. I'm just going to prescribe something else. Of course, the incidence of Stevens-Johnson syndrome fell correspondingly. That's good. But the incidence of Stevens-Johnson syndrome induced by alternate treatments went up. And the overall incidence stayed the same. So what that program shows is that there's an educational imperative that we're starting to meet. These are older data, and we all recognize that this is something that we have to do, and that's one of the reasons that I'm here. The other interesting thing about HLA-B 1502, B star 1502, is that it's predominantly in East Asia. But we see carbamazepine-induced Stevens-Johnson syndrome here. Munir, I think this may be the first paper in your CV, something like the second paper in your CV. Anyway, so he's been interested in carbamazepine for a long, long time. So this is an early paper, and then a New England Journal effort that he led, identifying a separate HLA allele, A star 3101, as the predominant allele in European ancestry populations. So again, these are the distributions, mostly blue dots, which means it's pretty rare, but occasional red dots down in South America, where it's even commoner. So ancestry really is important. And engaging the right ancestry and understanding who your patients are, it becomes important. So now people have started to look at whether pharmacogenetic testing makes a difference in outcomes. This is an early study from a Dutch group. They looked at variants in a gene called TPMT, thiopurine methyltransferase, and azathioprine, a drug that's used in inflammatory bowel disease. So loss of functional alleles in TPMT cause hematologic side effects. So they randomized a group of patients with inflammatory bowel disease to dose adjustment based on pharmacogenetics or not. And the first thing they did was look at whether they were influencing inflammatory bowel disease activity, because that would be another sort of spin-off. If you don't use the right drug, you have more inflammatory bowel disease. No difference there. And when they looked at hematologic adverse reactions across the board, there's no difference. But when you look only at people who carry TPMT variants, there was a huge difference. Well, huge, the numbers are small, but there's a big, big difference here. So obviously, if the pharmacogenetic strategy carries any weight, it's because it's going to be people who carry the variants in whom the adverse drug reactions are going to be avoided. And that's what these data suggest, but they're very small numbers. So there's a group called the Ubiquitous Pharmacogenomics Group, and they have been running a trial called PREPARE. PREPARE stands for that long thing down there. And it's basically a very, very large study conducted at 7 sites, each of them indicated by a star on this map, where the sites were randomized to pharmacogenetic testing or normal testing. And then after 18 months, they switched over to the other arm. So I don't know, and Liverpool was one of the sites. Munir will tell you which site he was and whether pharmacogenetics came first or the standard of care came first. The study started in 2017. Then there was this pandemic that got in the way. The study did not recruit its 8,100 subjects. It recruited about 6,700 patients. And it was powered to find a 30% difference in serious adverse drug reactions, from 4 to 2.8%. So the idea is looking for people not who have a little bit of a nosebleed, but people who get into the hospital because they're having an adverse drug reaction. Way back to the Liverpool data that I showed you earlier. One of the other things they did, which I think is actually really, really important, is everybody got a card in one of 7 languages, because there are 7 languages across this study. And the card lists their metabolizer status, lists whether there are drugs that they should or shouldn't be getting. There were 42 drugs that were evaluated in this study, and there's a QR code. So the pharmacist can look at the QR code, bring up the patient's records. And those data, the data from the study have been analyzed. They are in press in a major medical journal, and they're embargoed, so I'm not allowed to say anything more about that, except we should know the answer publicly in about 3 weeks. So strategies. This is a strategy document from the National Human Genome Research Institute, one of the major funders of genomic research in the United States. And I like to show it because it shows this intimate relationship between basic science and clinical science. And so what we do is, in basic science, we make discoveries, we implement, look at what happens when we implement, and then go back to the bench. So there are a couple of areas in pharmacogenetics that speak to this kind of algorithm. One of them is this. So I showed you a version of this before. Now, this time the poor and rapid metabolizers are on the other side of the axes, but the idea is the poor metabolizers in black are separable from the ultra rapids, and the other 2 groups may be overlapping a little bit. That's the effect of a single large effect size variant. But many drug responses are like this. They're a continuum, and the continuum reflects the fact that there are multiple genetic variants that go into determining whether somebody is at one end or another end of the spectrum. So that seems like an insurmountable problem, but it's not. In fact, if you look at clopidogrel responses, this is combining the effects of I think 11 variants in various genes that affect clopidogrel response. You can see that there's the more risk alleles you have, the higher the likelihood is that you'll have an undesirable antiplatelet effect. So that's one way of looking at it. The other way, which I find really interesting, is if you look, this is one of the newer, expensive, highly effective PCSK9 inhibitors for coronary artery disease, iliricumab. And if you look at people at baseline for risk of coronary artery disease based on what's called a polygenic risk score combining variants, you can separate people into low and high risk groups. And it's the high risk group they're high risk for the basic disease that you're treating who have the big effect of the drug. And the low-risk group don't have a big effect. So I think that has public health implications, and how it speaks to the relationship between drug effect and underlying disease susceptibility is an interesting outside question. I also showed you these data earlier, and it turns out that there are very, very high throughput methods being developed to look at not at 1 or 2 functional alleles at a time, but look at hundreds. These are data from Dick Winchelbaum at the Mayo Clinic, and they looked at several hundred CYP2C19 variants, and 121 and 109 CYP2C9 variants, and could score them. The abundance score gives a sense of pathogenicity. So many variants are normal, many variants are not. And these data may be important for implementation later. So let me just close with some thoughts. So I like the idea of making medical science work for everyone. I think that's a very compelling vision. We in the United States are trying to do that in something called the All of Us research program. In the United Kingdom, there's something called Our Future Health, which is going to recruit 5 million people. And I'm not going to talk about Our Future Health. I'll just show you a little bit of All of Us data. So we're aiming to get at least a million people, diverse backgrounds. Here are some of the numbers that we have so far. And our focus is on recruiting people who are traditionally underrepresented in biomedical research. So not a bunch of upper middle class white people, but a diverse population. And one of the other things that we're doing is we return data to participants. So we have started to return ancestry and trait data. The trait data are things like whether you don't like cilantro, whether you're lactose intolerant, whether you have hard or soft earwax. Those are things that people are interested in. They're genetically determined, and also their genetic ancestry. People are very interested in their genetic ancestry. We've offered it to 150,000 people to date, and 87 have chosen to receive some ancestry data at least. And then we are starting now to deliver what I would call harder genomic information. So hereditary disease risks across 59 genes known to have actionable variants that make people at higher risk for certain diseases, and then pharmacogenetics variants. We're returning variants in 7 genes, so starting slow, I think. And that's starting now, and there's a huge effort to create genetic counseling around this, and that's one of the key elements of this whole story. So, implementing pharmacogenomics should be simple. That's what Francis Collins said, it should be simple. And it's simple because these alleles are common, and they have pretty big effect sizes when they're there. There's not any angst, I call it angst, around whether to return results. If you ask people whether they want to know whether they're at high risk for breast cancer, or whether they're at high risk for coronary artery disease, or they're high risk for Alzheimer's disease, the answers are all over the place. Some people really want to know everything. Some people don't want to know anything at all. But then you ask them about pharmacogenetics, and they say, well, yeah, of course you should be doing that. None of those variants that I've talked about today have a link to underlying disease susceptibility. And electronic record systems can do this. It's hard, but they are getting better and better at doing this. So what are the objections? The cartoon obviously is meant to be a commentary on low-hanging fruit, in case anybody hadn't figured that out. So the alleles are common, but it's not so common to have a variant allele and to get the drug. So that's a rarer event. The assays can be complicated. Munir has actually developed some assays around HLA, which he makes work, but the rest of the world has trouble making it work. So I think you're really ahead of that. But some of the assays are not simple. There are many, many variants whose function we don't know, but we're attacking that problem. And then I think this is a big, big problem. Somebody has a variant in BRCA1 that confers susceptibility to breast cancer. They go to a counselor and a plan is made. Somebody has a variant in CYP2C19. They may or may not go to a counselor, but it might be a decade before they get prescribed Plavix. Or somebody has HLA A3101. It might be never, or it might be 15 years from now that they get Tegretol, carbamazepine prescribed. So whose problem is that? I think the patient has to own that. I think electronic record systems can help, but the patient, I think, is the only person who can own it. And I'm not sure how to solve that problem. And then healthcare providers, they're just learning how to do this, and the evidence base isn't there. So I think all those challenges are meetable. I mean, maybe the first way to solve a problem is to list the problem and then say it out loud, and then at least you could say, well, what am I going to do to fix this? But I think each one of those is addressable. So, and they're addressed in this document that Munir and others have prescribed. So I told Munir I took his 10 points and collapsed them into 4. So the idea would be to create a national program, not just at specialty hospitals, but widely and with support, with a continual evidence update, a big focus on education, on training, and on continued research. There are lots of research questions whose answers will inform how we do this, and then clear communication with physicians, with the public, with the patient populations, particularly all patient populations, including diverse ancestries. So I told Munir I was stop with a slide that he'll be surprised by. So the last time I was in Liverpool, this was the newspaper headline. I thought this was really cool. It said genetic testing on the NHS. And the second bullet point is patients' lives will be transformed as new service will allow personalized treatments. That was the headline, 3 or 4 years ago. So I think the time for the will is gone. I think it's now, not will. So thank you so much for the opportunity to expound, and I'm happy to take questions.